Hello, everyone. This is Dr. Kellen Cavanero, and welcome to another episode of Inflammatory Content, a podcast about immunology and biomedical research more broadly. It is normally my job to interview world-class researchers to broadly communicate the cutting-edge work going on in their laboratories and to get to know the people behind the science. Today, we're going to do a journal club, but it will not be your run-of-the-mill journal club. Everyone who knows me knows I am always searching for the latest technology that can increase my efficiency. Lately, I have been especially attuned to the newest developments in artificial intelligence, or AI. Well, I recently discovered a new tool by Google called NotebookLM that creates audio overviews of documents. What this means is that it will, within minutes, create a journal club podcast if you upload a research article. It is absolutely bananas. There are a few minor instances where the speech is buggy and words are mispronounced, but overall, I am super impressed with how well it communicates research findings. Very accessible and high fidelity. So today, I bring to you an experimental AI-generated journal club on my two latest research articles. The first article was published in the Journal of Experimental Medicine in early 2024, and the second was published as a preprint, meaning not yet peer-reviewed, on bioRxiv in late 2024. So sit back, relax, and let's dive into some science. And if you enjoyed the show, please consider sharing the episode, leaving a review, or donating to support my work. Thanks for listening.
Welcome back, everyone. So today we're going to be doing a deep dive into some research that you all sent over, and it looks really fascinating. It's all about how our skin fights off infections, and it seems like there's a lot more to it than just being a physical barrier.
Oh, absolutely.
Yeah.
Yeah, there's a lot going on beneath the surface, as we'll see.
So maybe you can give us a little preview of what the big discovery is here.
Yeah, so this research is really focused on fibroblasts.
Okay.
And you might remember these as kind of like the structural cells that make up like the scaffolding of our skin.
Right. Yeah, kind of like the building blocks.
Yeah, exactly. But it turns out they're not all just these passive support beams.
Okay.
This study really delves into this, and it reveals a special subset of fibroblasts called CXCL12 plus fibroblasts.
CXCL12 plus?
Yeah, it's a mouthful.
Okay.
But these fibroblasts, they play a really key role in like orchestrating the skin's immune defense.
Wait, so I thought fibroblasts were just like scaffolding, like you were saying. How can they be so like diverse and specialized?
Yeah, that was kind of the prevailing view for a long time.
Okay.
But thanks to new techniques like single-cell RNA sequencing, scientists can now analyze individual cells in much greater detail.
Oh, wow.
It's pretty amazing.
Yeah.
It's like taking a census of each cell
Wow.
and figuring out its unique function based on the genes that it's expressing.
Okay.
And that's really how they uncovered these CXCL12 plus fibroblasts.
So what makes these CXCL12 plus fibroblasts so special?
Yeah.
What do they do that other fibroblasts don't?
Well, these fibroblasts are kind of like the communication hubs of the skin's immune system.
Okay.
They're particularly good at talking to neutrophils.
Neutrophils.
Yeah, these are white blood cells that act like first responders, rushing to the site of infection to fight off invaders.
The front line.
Exactly.
Okay, so I'm curious to know what happens when our skin actually gets infected.
Yeah.
How do these CXCL12 plus fibroblasts sort of spring into action?
So the researchers, they actually looked at Staphylococcus aureus skin infections, which, these are a common cause of those nasty boils and abscesses that we sometimes get.
Yeah, nobody else is.
No, not fun. But what they found was that when skin is under attack by these bacteria, the CXCL12 plus fibroblasts, they start producing a whole bunch of chemokines.
Chemokines now. There's
Signaling molecules. So they're like these little homing beacons that attract neutrophils to the site of infection.
Oh, okay.
So it ensures a swift and targeted immune response.
So these fibroblasts are basically sending out an SOS to all the neutrophils in the area, like, hey, we need you over here. We've got a situation.
Exactly. It's like a little 911 call.
Oh, like it.
And they don't just send like a general alert either. They seem to fine-tune the response based on the specific threat.
Oh, wow. That's really interesting. So how do these fibroblasts know when to sound the alarm?
Right.
Like, what triggers them to start producing these chemokines?
Yeah, so they're incredibly sensitive to certain signals in their environment, and one of the key triggers that they discovered was IL-17. And this is a powerful signaling molecule often produced during infections and inflammation.
So IL-17 acts like an amplifier.
Uh-huh.
It's boosting the signal from these fibroblasts.
Exactly. When IL-17 is present, the CXCL12 plus fibroblasts produce even more of those neutrophil-attracting chemokines.
Wow.
And it doesn't even stop there. They also discovered that another signaling molecule, TNFi,
Okay.
this works in synergy with IL-17 to further amplify the response.
Wow.
It's like a 1-2 punch
Wow.
that ensures a rapid and robust immune response.
Okay, so we've got these specialized fibroblasts sending out an SOS signal.
Yeah.
It's amplified by IL-17 and TNFi calling in the neutrophil troops. But how is all of this controlled?
Yeah.
There must be some kind of internal switch that regulates this whole process.
You're absolutely right. And that's where a key transcription factor called NFKBIZ comes in.
NFKBIZ.
Yeah, I know it's a mouthful.
Yeah, that sounds complicated.
It does have a rather technical name. But essentially, NFKBIZ act like the master regulator.
Yeah.
Making sure the CXCL12 plus fibroblasts produce the right amount of chemokines at the right time.
Okay.
And the researchers found that if they disrupted NFKBIZ, the whole defense system went haywire.
Oh, wow.
Yeah. The fibroblasts didn't respond properly to the infection,
Okay.
and the neutrophils weren't recruited effectively.
Wow. So this NFKBAZ is crucial for making sure this whole intricate defense system works as intended.
Yeah, it's really a linchpin.
This is really groundbreaking stuff. It makes you realize just how much we're still learning about the complexity of the immune system.
Absolutely.
Yeah.
And what's even more exciting is that this discovery has broader implications beyond just bacterial infections.
Oh, really?
Yeah.
Tell me more. So we've been talking about how these CXCL12 plus fibroblasts help fight off bacterial infections.
Right.
But you mentioned there were broader implications. So what else did the researchers find?
Well, they wondered, if these fibroblasts might be involved in other skin conditions, not just these acute infections. So they decided to investigate their role in psoriasis.
Psoriasis. Now, that's a chronic skin condition, right? It's not caused by bacteria, is it?
Right. Psoriasis is an autoimmune disease where the immune system mistakenly attacks the skin.
Okay.
But while the root cause is different from a bacterial infection, it still involves a lot of inflammation
Yeah.
and the recruitment of immune cells to the skin, including those neutrophils we've been talking about.
So they basically used psoriasis as a model to see if these fibroblasts were involved in kind of a broader range of skin immune responses.
Yeah, exactly.
Interesting. So what did they find?
Well, they used a mouse model of psoriasis. And to their surprise, they found that those CXCL12 plus fibroblasts were activated in this model too.
Oh, wow.
It seems like they're not just specialized for fighting bacteria.
Yeah.
They're involved in a more general inflammatory response in the skin.
So these fibroblasts are like the Swiss Army knife of skin immunity.
Yeah, that's a good way to put it.
That's pretty cool. But did they find any evidence of this in humans? Because mouse models are great and all, but they're not always a perfect reflection of human biology.
That's a great point. To see if this held true in humans, they analyzed skin biopsies from people with psoriasis.
Okay.
And they were able to identify these same CXCL12 plus fibroblasts in the human psoriasis skin.
So that's pretty compelling evidence that this isn't just a mouse thing.
Yeah, for sure.
Did they find anything else in the human studies?
Yes. They found that the activity of these fibroblasts was reduced when the patients were treated with IL-17 inhibitors.
Hold on. Remind me, what are IL-17 inhibitors again?
Oh, yeah. So these are a class of drugs that are used to treat psoriasis. Remember how we talked about IL-17 amplifying the fibroblast response?
Right.
Well, these drugs block IL-17, so they're dialing down that inflammatory signal.
Ah, okay. So by blocking IL-17, these drugs are indirectly affecting the CXCL12 plus fibroblasts and leading to less inflammation in the skin. That's a really interesting connection.
It is. It really highlights the potential importance of these fibroblasts as therapeutic targets. If we could find ways to specifically modulate their activity, we might be able to develop new treatments for a whole range of inflammatory skin conditions.
That's a pretty big deal.
It is.
So what does all of this mean for, like, the average person dealing with skin issues?
Well, it's still early days for this research.
I'm sure.
But it definitely opens up new avenues for understanding and potentially treating skin diseases.
Okay.
It also underscores just how complex the skin's immune system really is.
It's not just a passive barrier.
Right.
It's like a dynamic ecosystem with all these different specialized cells working together to protect us.
Exactly. And these CXCL12 plus fibroblasts are turning out to be more important than we ever realized. They're like the unsung heroes of skin defense.
I like that. So I have one more question.
Yeah.
This research focused on the skin.
Right.
But could similar fibroblasts be at work elsewhere in the body? Could they be involved in immune responses in other organs?
That's a fantastic question.
Yeah.
And it's something that the researchers themselves brought up in their paper.
Oh, really?
Yeah, they speculated that similar types of fibroblasts might exist in other tissues,
Okay.
especially those that are constantly exposed to the outside world,
Right.
like the lungs or the gut.
That makes a lot of sense. It's like this research is opening a door to a whole new world of immune cell interactions. So much more to explore.
Absolutely. And it's a great reminder that there's always more to discover.
Yeah.
That's what makes science so exciting.
Yeah.
There are always new mysteries to unravel, new connections to make new insights to be gained.
Absolutely. Okay, before we move on, let's just take a moment to summarize what we've learned about these incredible CXCL12 plus fibroblasts.
Okay.
All right, so just to recap, we learned that these CXCL12 plus fibroblasts are way more than just those structural cells we thought they were.
Yeah, definitely not just scaffolding anymore.
Right. They're key players in our skin's immune response, and they're not just sitting there passively either.
No, they're actively communicating with other immune cells, like those neutrophils,
Right.
orchestrating this whole defense against threats,
Yeah.
like those Staphylococcus aureus bacteria that cause skin infections.
And I think what I find particularly interesting is that they're not limited to dealing with just infections. They seem to play a role in a wider range of inflammatory processes in the skin, like we saw with the psoriasis research.
Absolutely. And we learned about how IL-17 and TNF,
Right.
those signaling molecules, they act like amplifiers boosting the fibroblasts' response.
Right.
It's really a finely tuned system.
Yeah. And then there's that master regulator, NFKBIZ, making sure everything runs smoothly. It's amazing how much is going on beneath the surface of our skin.
It really is.
Yeah.
And the fact that scientists are uncovering these intricate details at the cellular level, it opens up so many possibilities for new treatments.
Yeah.
Imagine being able to target these fibroblasts directly to either enhance the immune response when we need it or dampen it down when it's causing problems like in psoriasis.
Exactly. The therapeutic potential is huge. This deep dive has definitely given me a whole new appreciation for the complexity of the immune system.
Me too. And it's a good reminder that we still have so much to learn. Every new discovery seems to open up even more questions.
No, true. Well, on that note, we'll leave you with a final thought to ponder. We've been talking about the skin,
Right.
but as we touched on earlier, could these types of fibroblasts be involved in immune responses in other parts of the body? What about the lungs or the gut or other places where our bodies interact with the outside world?
It's a fascinating question to consider. And it seems like we've only just scratched the surface of understanding the full impact of fibroblasts on our immune system as a whole. Perhaps in the future, we'll be doing a deep dive into CXCL12 plus fibroblasts in the gut or the brain. Who knows what we'll find?
The possibilities are endless. Thanks for joining us on this deep dive. We'll catch you next time for another exploration of cutting-edge research.
Welcome back, everybody, for another deep dive. Today, we are going to be tackling Sweets syndrome.
Okay.
You've probably heard of it. It's an uncommon skin condition, marked by these painful nodules and plaques.
Yeah, definitely.
But the thing is, it's a bit of a medical head scratcher.
It is. It really is. We know there's this massive influx of neutrophils into the skin.
Right.
But the underlying cause is it's remained elusive.
Yeah.
And then the standard treatment, systemic steroids, they often come with a whole bunch of unpleasant side effects.
Of course.
So the research you shared dives deep into this molecular landscape of Sweets syndrome.
Yeah.
And it's comparing it to other skin conditions to kind of uncover potential new treatment strategies.
Yeah, and they use some pretty high-tech methods to do this.
They do.
Single nucleus RNA sequencing, bulk RNA sequencing,
Right.
and spatial transcriptomics.
Wow.
Can you break those down for me a little bit? What exactly are these techniques and why
Yeah.
are they so important for this kind of research?
It's like taking a high-resolution snapshot of what's happening inside individual cells.
Oh, wow.
So single nucleus RNA sequencing allows scientists to analyze the gene expression of each cell in a tissue sample, giving them incredible detail.
Wow.
Bulk RNA sequencing gives you a broader overview of the gene expression across all the cells in a sample.
Gotcha.
And then spatial transcriptomics adds another layer. It maps where those gene expression patterns occur within the tissue.
So it's like having a GPS for cells.
It is. It's like having a GPS for cells.
That's amazing.
Yeah.
So it's not just about identifying the players,
Right.
but understanding their roles and relationships
Exactly.
within the larger ecosystem of the skin.
A whole environment.
Yeah.
What did these techniques reveal about Sweet syndrome?
So one of the most striking findings was this prominent interferon signature.
Okay.
Specifically type I interferon.
Okay.
And that was unique to Sweet syndrome.
Interesting.
This wasn't really present in the other skin conditions they studied.
Huh. So interferon, that's something scientists, I feel like, already suspected.
Yeah.
Or was this like a totally unexpected finding?
I mean, while interferon is well known for its role in antiviral responses,
Right.
its involvement in Sweet syndrome wasn't fully appreciated until recently.
Okay.
So this research really strengthens the case for interferon being a key driver
Gotcha.
of that inflammatory process.
So interferon is on the scene.
It is.
But where did the fibroblasts come in?
Right.
I always thought of them as like the scaffolding of the skin,
Yeah.
just like providing that structural support.
Right.
How are they involved in inflammation?
Yeah, so that's where it gets really interesting.
Okay.
Traditionally, fibroblasts were seen as these passive players, but this research, along with a growing body of evidence, reveals that they have a much more active role, particularly in inflammatory conditions.
Okay, so they're not just sitting there holding things together.
No, they're not.
What are they actually doing?
Think of them as the communication hubs of the skin. So when activated by type I interferon, fibroblasts start releasing these things called chemokines.
Okay.
And those are signaling molecules that attract other cells, specifically neutrophils, to the area.
So they're calling in the troops.
Exactly.
Yeah.
They're calling in the troops.
I like it.
And the neutrophils being the first responders of the immune system,
Right.
they rush to the scene ready to fight.
Yeah.
But in Sweet syndrome, this response is exaggerated,
Okay.
leading to that excessive inflammation and those painful skin lesions.
That makes sense. But why is Sweet syndrome different from other conditions like psoriasis,
Yeah.
where we also see, a lot of neutrophil activity?
That's a great question. So while both conditions involve neutrophils, the underlying mechanisms seem to differ.
Okay.
So in psoriasis, the inflammatory molecule IL-17 plays a central role.
Right.
But in Sweet syndrome, IL-17 isn't a major factor.
Interesting.
Instead, this research highlights the role of those interferon-activated fibroblasts in driving that neutrophil influx.
So it's fascinating how one type of cell can have such a dramatic impact
I know, it's amazing.
on the overall inflammatory process.
Yeah.
But what about those other interferon-activated cells mentioned in the research?
Right.
Keratinocytes, myeloid cells, lymphocytes.
Yeah.
Are they also contributing to the inflammation in Sweet syndrome?
It's certainly possible, and that's something researchers are actively investigating.
Okay.
The complexity of the immune system lies in this intricate interplay between all the different cell types.
Yeah.
So this research has shown a spotlight on fibroblasts.
Right.
But understanding the contributions of these other cell populations is going to be crucial for developing a complete picture of the disease.
For sure. So speaking of other cells, the research briefly touched on immune-acting fibroblasts, or IAFs, and their role in other conditions.
Yeah.
Can you elaborate on that a little bit? How do IAFs differ from the fibroblasts involved in Sweet syndrome?
IAFs are a specialized subset of fibroblasts, and they've been shown to play a role in various inflammatory and autoimmune diseases, including psoriasis. So while both IAFs and the interferon-activated fibroblasts in Sweet syndrome contribute to inflammation, they do so through different mechanisms. So for example, in psoriasis, IAFs are often activated by IL-17, which, as we discussed, isn't a major player in Sweet syndrome.
Right. So while there are some parallels, the specific triggers and pathways seem to be distinct.
Definitely.
This research also mentioned a 2024 study about interferon and liver cancer cells.
Yes.
Why was that study included?
Okay.
And what's the connection to Sweet syndrome?
So that study, while focusing on liver cancer, revealed a fascinating parallel with Sweet syndrome.
Okay.
So researchers found that type I interferon could induce the secretion of CXCL8, which is a potent neutrophil chemokine in liver cancer cells. So this finding further supports the idea that interferon can drive neutrophil recruitment in various contexts, even outside of traditional inflammatory settings.
So even though the context is different, liver cancer versus a skin condition,
Totally.
the underlying mechanism involving interferon and neutrophil recruitment seems to be shared.
It does seem to be shared.
That's a pretty remarkable connection.
It is.
But what does this mean for potential treatments?
That's where things get really exciting.
Ooh.
And that's what we'll delve into next.
All right, I'm hooked. Let's move on to part 2.
So, one of the most perplexing things about Sweet syndrome is how it can be triggered by so many different things. Infections, cancer, even pregnancy.
Yeah.
It almost seems random.
Yeah, it's hard to imagine how such diverse triggers could lead to the same condition.
Right. And that's where this research offers a new perspective.
Okay.
It suggests that while those initial triggers may differ,
Yeah.
they might all converge on this common pathway,
Right.
the interferon pathway.
I'm following you. Explain that a lot.
Okay, so
Yeah.
think about it. Interferon is this key player in our immune response, not just to viruses, but to other threats as well,
Okay.
like bacteria and cancer cells.
Uh-huh.
And guess what? Infections, cancer, pregnancy,
Yeah.
they can all kickstart interferon production.
So you're saying that even though sweet syndrome can have all these different starting points,
Right.
they might all ultimately lead to the activation of the interferon pathway.
Precisely.
Okay.
And to make it even more compelling,
Yeah.
the research cites numerous case studies where people actually developed sweet syndrome after receiving interferon therapy.
Wow.
It's like a direct link.
Yeah, that's a pretty strong correlation.
It is.
It really points to interferon as a central player.
Yeah.
But how do those fibroblasts we talked about earlier fit into this bigger picture?
Right. That's the million-dollar question.
Yeah.
So it seems that interferon might be the initial trigger,
Okay.
setting off this whole cascade of events that ultimately leads to fibroblast activation and all the downstream inflammation.
So it's like a domino effect.
Something triggers interferon,
Yep.
which activates fibroblasts,
Mm-hmm.
which call in the neutrophils,
Right.
and boom, you've got sweet syndrome.
That's a great analogy.
I try.
And remember those two distinct fibroblast populations we discussed?
Yes.
The ones located in different layers of the skin,
Right.
producing different chemokines.
Right.
Well, this research suggests they might be playing distinct roles
Okay.
in orchestrating this whole neutrophil invasion.
Okay, so maybe the fibroblasts near the surface are sending out the initial come hither signal to the neutrophils circulating in the blood.
Mm-hmm.
And then the deeper fibroblasts might be guiding the neutrophils to their final destination,
Right.
telling them where to go, what to do once they arrive.
That's a fascinating hypothesis, isn't it?
It is.
It suggests this level of coordination and complexity
Yeah,
that we hadn't really appreciated before.
totally.
And it highlights how important spatial context is when we're trying to understand how these cells interact
Right.
and communicate. Thinking about these fibroblasts, it's fascinating how cells we usually associate with structure
I know.
can play such a central role
Yeah.
in inflammation.
Yeah,
It really makes you realize how interconnected everything is within the body.
it is.
But I'm still curious about those other interferon-activated cells mentioned in the research.
Okay.
The keratinocytes, myeloid cells, lymphocytes.
Right.
Could they be playing like a supporting role
Potentially.
in this inflammatory cascade?
It's certainly a possibility. While this research focuses on fibroblasts, it's important to remember that they're not acting in isolation.
Of course.
The immune system is this complex network of interacting cells,
Right.
and understanding the contributions of these other cell populations is going to be crucial
Yeah.
for developing that complete picture of sweet syndrome.
It sounds like there's still a lot to uncover.
There is.
But the fact that we're starting to identify these key players
yeah,
and their interactions is a huge step forward.
absolutely.
Yeah.
And remember, this research isn't just about sweet syndrome.
Right. The insights we're gaining about interferon,
Mm-hmm.
fibroblasts, and spatial organization
Yeah.
could have implications for understanding and treating a whole range of inflammatory and autoimmune diseases.
That's a really exciting prospect. If
Right.
we can figure out how to control interferon signaling
Yeah.
and fibroblast activity, could we potentially develop new treatments for conditions like lupus,
It's possible.
where we also see interferon and neutrophils causing trouble?
Yeah, it's certainly a possibility worth exploring.
Yeah.
Imagine being able to selectively dampen inflammation in specific tissues
Right.
without those side effects of systemic steroids.
Right.
That would be a game changer for so many people.
It's incredible to think that a deep dive into a rare skin condition could unlock such profound insights into the workings of our immune system.
The human body is amazing.
It really is. But before we get too carried away,
Yeah,
it's important to acknowledge that no study is perfect.
you're absolutely right.
Uh-huh.
What are some of the limitations we should keep in mind? Well, one thing to note
Okay.
is that the single nucleus RNA sequencing analysis only included one sample from a patient with pustular psoriasis.
Right.
While it was valuable as a comparator to sweet syndrome,
Yes.
we can't draw any firm conclusions about pustular psoriasis more broadly
Right.
based on just one sample.
Of course.
That makes sense. It's always important to remember that scientific findings need to be replicated and
Yeah.
validated in larger studies before we can generalize.
Absolutely.
And while the spatial transcriptomics data included 2 additional pustular psoriasis samples,
Right.
which supported the initial findings,
Yes.
it's still a relatively small sample size.
It is a small sample size.
So we need to be cautious about overinterpreting the results,
Right.
especially when it comes to comparing sweet syndrome to other conditions.
Exactly. But even with those limitations,
Yeah,
this research is incredibly valuable.
for sure.
It provides a solid foundation for future studies
Right.
and opens up this whole new avenue for exploring potential treatments.
Totally.
Yeah.
And let's not forget the significance of those detailed cellular maps generated by this research.
Oh, yeah.
I mean, they're an invaluable resource
They are.
for scientists studying these diseases.
Huge. Speaking of treatments, the research mentioned JAK-STAT inhibitors
Yes.
as a potential therapeutic avenue.
Right. Can you remind me how those work
Yeah.
and why they hold promise for treating sweet syndrome?
So JAK-STAT inhibitors are a class of drugs that block the signaling pathway activated by interferon.
Okay.
And remember how we talked about interferon being that initial trigger that sets off the inflammatory cascade?
Right.
Well, JAK-STAT inhibitors essentially cut off the communication from interferon,
Okay.
preventing it from activating those fibroblasts
Right.
and triggering that whole chain of events.
So it's like hitting the off switch on the interferon signal,
That's-
preventing the downstream inflammation.
Mhm.
And the fact that there are already FDA-approved JAK-STAT inhibitors for other conditions
Right.
is encouraging, right?
It is. It means we already have a head start in
Right.
terms of safety and efficacy data.
Yeah.
Of course, more research is needed to confirm their effectiveness, specifically for Sweets syndrome,
Sure.
but the potential is certainly there.
This is all so fascinating.
It is.
We've gone from painful nodules to cutting-edge technology,
Yeah.
from rogue fibroblasts to promising new therapies.
It's quite a journey.
It's amazing to see how scientific inquiry can lead to such incredible insights and potential breakthroughs.
Yeah.
But I'm still trying to wrap my head around how all of this actually plays out at the cellular level.
Let's shift gears then and
Awesome.
try to visualize what's happening inside the body.
Sounds good. Let's move on to part 3. All right, so we've covered a lot of ground, but can we try to visualize this whole process? Like what's actually happening at the cellular level in someone with sweet syndrome?
Sure. So let's paint a picture. Imagine you have a trigger. Maybe it's a bacterial infection or even certain medications. This trigger sets off an alarm in your body, causing it to release this surge of interferon.
Okay, so interferon is the first responder.
Right. What happens next is that interferon starts binding to receptors on various cells in your skin,
Okay.
including those fibroblasts we've been talking about.
Right.
It's like a wake-up call telling them to shift from their normal structural role to a more inflammatory one.
So the fibroblasts get the signal and they're ready for action.
Right.
They do.
Remember those chemokines we discussed? The chemical signals that attract other cells?
Yes.
Well, the activated fibroblasts start producing a whole bunch of chemokines,
Okay.
specifically the ones that attract neutrophils.
Uh-huh.
It's like sending out an SOS summoning the troops.
And the neutrophils being the first responders of the immune system,
Right.
they answer the call and rush to the site of the action.
Exactly. They flood into the skin ready to fight.
Mm-hmm.
But here's the catch. In Sweets syndrome, this response is out of proportion to the actual threat. It's like sending in an army to deal with a minor skirmish.
So you have all these neutrophils going into overdrive,
Yeah.
releasing inflammatory molecules,
Right.
causing the redness, swelling, and pain that's typical of sweet syndrome.
That's right. And to make matters worse, those neutrophils can also release something called neutrophil extracellular traps,
Okay.
or NETs for short.
Nets. I haven't heard of those. What are they?
They're basically webs of DNA and proteins that neutrophils expel
Okay.
to trap and kill pathogens.
Gotcha.
But in Sweets syndrome, these NETs might actually be contributing to the inflammation and tissue damage.
Interesting. So instead of helping, they might actually be making things worse.
That's the current thinking. They're like friendly fire in this scenario.
Yeah.
And to add another layer of complexity,
Yeah.
the research also mentioned plasmacytoid dendritic cells, or PDCs.
Right. Those were mentioned briefly. How do they fit into this puzzle?
Well, PDCs are major producers of interferon.
Okay.
And when they encounter those neutrophil nets, they get activated and start producing even more interferon.
So it's like a vicious cycle.
It is.
Interferon activates fibroblasts, which
Yeah.
attract neutrophils,
Mm-hmm.
which release nets,
Right.
which activate PDCs to produce more interferon and so on.
Exactly. This positive feedback loop could help explain why sweet syndrome can be so persistent and difficult to treat.
Wow. It's amazing how all of these different cells and molecules are all interconnected,
I know, it's fascinating.
working together or sometimes against each other
Right.
to maintain this delicate balance.
It really highlights the complexity of the immune system.
It does.
And while this research has shed light on a potential key mechanism in sweet syndrome, it also opens up this whole new set of questions.
Like what?
Well, for example, what are the specific signals that activate those different fibroblast populations?
Right.
Can we develop therapies that target those specific signals?
Yeah.
What role do those other interferon-activated cells, the keratinocytes, myeloid cells, lymphocytes, play in the overall inflammatory process?
So many questions.
So many questions.
It's exciting to think about all the potential avenues for future research.
It really is. And with advances in technologies like single-cell sequencing and spatial transcriptomics,
Right.
we now have the tools to start answering these questions.
Well, this has been an incredibly insightful deep dive.
Yeah.
I feel like I have a much better understanding of Sweets syndrome now. Not just the what, but the why and the how.
I'm glad to hear that. And remember, this isn't just about a rare scare condition.
Right.
The insights we're gaining about interferon fibroblasts and the immune system
Yeah.
have broader implications for understanding and treating a wide range of inflammatory and autoimmune diseases.
Absolutely. For our listeners who want to explore further, we've included links to the research papers in the show notes. And if you're interested in learning more about the fascinating world of single-cell analysis and spatial biology, we'll be doing a deep dive into those topics in a future episode. So stay tuned for that. Thanks for joining us on this deep dive. Until next time, keep exploring, keep learning, and keep asking questions.
See you next time.
And that concludes today's fully AI-generated episode. Pretty crazy, right? I know it's not perfect, but I hope you all still found it helpful. If you did, please consider sharing the episode with your friends and colleagues, writing a review, or donating to support the show. You can find all relevant links in the show notes. And please reach out to me with any questions or comments. I would especially like to know if you want to hear more AI-generated podcasts and also how you may be using AI to enhance your science.